How's it going, everyone? Today we'll be discussing how skeletal muscle fibers and other different types of muscle fibers get ATP. And so this first part will be a review, but then we'll talk a little bit more about muscle fatigue and then the things that help us generate more force out of our muscle fibers. Like we said earlier, myosin needs ATP in order to have these contractions taking place. And so our skeletal muscle cells need ways for us to get that ATP. And so we talked a lot about this in the metabolism chapter, so I'm not going to beat it over the head too much here, but we will go into the different ways our muscles get ATP. The first way is just using our ATP stores. So whatever ATP is available within the skeletal muscle cell, within those first 2 to 3 seconds, we're going to have enough to produce contractions within those time periods, but those stores run up really quickly. So we're going to need to have other molecules and other materials which are going to help us create ATP within our skeletal muscle cells. So first we're going to use ATP, but our second source of ATP in our skeletal muscle cells is going to be creatine phosphate. And so what's going to happen is that you're going to have a enzyme called creatine kinase, which is going to allow creatine phosphates within our cells to donate a phosphate group to ADP in order to get ATP. So this is our second way, basically, of storing, our next reserve of ATP. So whenever we need more ATP, we just, cleave off the phosphate group, give it to ADP. Now we have ATP. And so you're probably familiar with creatine. It's just giving us basically the ability to store more ATP. And so, if we need to store it, the reaction goes in the forward direction. If we need to use it, the reaction goes into the reverse direction. But again, there's not much, as far as storage is concerned, much energy that we could get from creatine phosphate. It's only going to supply us with around 30 seconds of ATP energy once we've used our ATP stores. So we're going to need to have other ways, more complex ways, in order to produce ATP for skeletal muscle fibers to use. In addition to our ATP stores and our creatine phosphate stores, which don't really last too long, we also can use glucose and fatty acids in order to make ATP. And we discuss that glycolysis is going to break down glucose in order for us to get quick ATP. And then lipids can be broken down further as well through aerobic respiration in order to generate ATP. But it's going to depend on how much we use of each, depending on the intensity of the activity. So we can see in the graph here that at lower intensities that we are going to use more free fatty acids. And then as we increase in exercise intensity, as we approach VO2 maximum, that's where we're going to shift more towards anaerobic metabolism, because we're not able to really keep up with the rate of oxygen required to carry out the exercise. And so once that happens, once you're not getting as much oxygen, but that ATP demand's going up, that's when you're starting to shift to more anaerobic metabolism. And so as intensity of exercise gets harder, we tend to gravitate more towards using glycolysis, and that's going to in turn produce lactic acid. But at lower intensities, we will use more so free fatty acids in order to generate our ATP. You are going to rely more on glycolysis when your demand for work in ATP exceeds the amount of oxygen available to make that ATP. So if you think about when this occurs, right, you're exercising, when would you rely more on glycolysis? Well, we just said at high intensities, right? We have a high intensity, let's say 85% above our VO2 max. We're sprinting, we're lifting, near maximal weights over and over again. Well, we are having a high demand of work, and we need to produce high levels of muscle contraction. There's a lot of motor units that are being activated and generating a contraction, so they need a lot of ATP. And glycolysis gives you a way to quickly produce ATP without high levels of oxygen. But there is a downside to this, because if your work demand's so high and these oxygen levels cannot keep up, you're going to produce lactic acid, as we talked about. And so that pyruvate, it turns into lactic acid instead of going through the Krebs cycle and electron transport chain. So this lactic acid, it actually compromises performance, and that's why you can't keep up high intensities for that long. Another situation where you would use more glycolysis to generate your ATP would be at altitude. Because when you think about it, there's less pressure of oxygen at higher altitudes, and therefore then there's less available oxygen, and you need to rely more on anaerobic metabolism. And some coaches take this into consideration. Let's say, for example, you had an event in Denver or Mexico City. Well, you're going to need to adjust and train in order to make sure that you rely more on those, anaerobic metabolic pathways to generate that ATP. So many coaches take into consideration all these different factors while training to know which style of training and what intensities are best based on their athlete's goals. You are going to rely more on aerobic respiration for longer bouts, less intense in bouts of physical activity. So, if you want to make a comparison, a marathon runner would, of course, rely more on aerobic respiration in their event, and a 400-meter sprinter would much more rely on, glucose metabolism during their events. And so, you have all these different sports which sometimes, include a combination of both of these pathways. Certain sports have quick, little bursts of intense movement. I could, think of like soccer or basketball, because, not only do you have to last the whole time running up and down the court or up and down the field, but also you're going to have these quick bouts of agility, and explosiveness. So it's really important when you train or you're working with athletes to realize what elements are a part of their sport, what kind of metabolism and what kind of cardio are they going to need in order to accelerate well in their sport, and to gear a program based on those things. Muscle fatigue is going to be a temporary decline in muscle force. So when you think about it, if you are exercising for an extended period of time and you're working the same muscle groups over and over again, you're likely not going to be as strong at the end of the workout as you are in the beginning of the workout. And now muscle fatigue can be brought about by multiple factors. Central fatigue is one reason why, and that has to do with alterations in our central nervous systems that lead to differences in motor unit recruitment and activation. And a lot of times it's brought about by different signaling, different levels of hormones like serotonin, dopamine, and norepinephrine. Muscle fatigue can also be brought about by local factors as well. To start, if you don't have enough ATP to generate high levels of force, you are going to fatigue. And so you're going to need a bunch of ATP in order to keep up the amount of work during exercise. And so that's why oxygen and glycogen and glucose, it's also important, because that's how you're providing the muscle the ability to generate contractions. And then things like calcium, of course, are going to be important. That level of calcium within the cytoplasm and the amount that binds to troponin is going to determine how many cross bridges are formed. So if there's declines in that, your muscles can fatigue as well. Acetylcholine is the neurotransmitter which helps start the whole excitation, contraction, coupling process. And this last one's a little bit more debated than you would think. The role of lactate and lactic acid in muscle fatigue has sort of been debated amongst different groups of researchers, but I will throw it in there, and I will say that there is evidence that definitely muscle strength goes down as lactic acid does build up. That is for certain, but people debate whether it's because of the calcium sensitivity of the fibers is affected by pH. But anyway, yes, that lactic acid is producing some sort of fatigue because as lactic acid does build up, the force does tend to decline. Once you start to work out, you are relying on all these anaerobic, all the pathways that don't involve oxygen in order to get this ATP, right? So you're using these stores when you first begin to exercise. And then as you continue along, as long as the exercise is prolonged for a certain amount of time, then that oxygen will sort of start to kick in. That's probably a horrible way to describe it, but it will start to help create more ATP through aerobic mechanisms, right? But once you stop exercising, you need to make up for all of those stores that you lost. And in order to do that, we're going to consume more oxygen after exercise. And so that phenomenon is called exercise post-oxygen consumption, or EPOC. And the reason why we're consuming more oxygen, after the fact, after the workout, is because we're in an oxygen debt. Again, we are using, once you start to work out and you start moving, that ATP demand jumps. So we need to start using all these stores and using all the available oxygen that we have in that second. And then later after the exercise, then we start to make up for it by increasing our respiration. And so it's important that oxygen will be used to help convert lactic acid to glycogen for tissue repair and then resynthesizing creatine phosphate. The amount of tension that a skeletal muscle fiber can produce is based on a few different things. One is going to be the frequency of action potential sent by the alpha motor neuron. Two is going to be the length of the fiber and the speed that the fiber is contracting. And then we're also going to see the differences in force produced by skeletal muscles under different types of contractions. When a alpha motor neuron fires one action potential and those acetylcholines bind to receptors on the skeletal muscle fiber, it's going to cause a twitch, and that's when the muscle has a singular contraction and then a singular relaxation. But if we want to increase the amount of tension that this fiber could produce, what we could do is send another action potential before it relaxes in order to increase the amount of calcium in the fiber and then therefore increase the amount of force produced by that fiber. So here, you see again a single action potential and a single twitch. But if we sent another action potential here, well then now that tension is going to increase. And as we increase the frequency of these action potentials, we get a summation of their forces. And so this will go higher and higher. But now these are all unfused contractions. You could tell by the little dips that are still occurring, as this fiber is contracting. But if you send action potentials at a high enough frequency, fast enough, then these twitches are going to fuse, and that's what we call a fused titanic contraction. And this is how most of our skeletal muscle movements are day to day, right? We rely on these titanic contractions for the muscles to produce force. We signal high amounts of action potential down that alpha motor neuron, and then once we're done with the movement, those action potentials stop, and then muscles relax. So this is what creates smooth movements, because if we were trying to lift our arm up, we wouldn't want those twitches occurring. So by increasing the frequency of those action potentials, we're able to produce higher amounts of force and smooth out contractions. Here we can see the relationship between length, velocity, and tension. And so when you have your fiber, or let's say your sarcomere, right? As you increase the length of your sarcomere and then do an isometric contraction, you're going to see that's going to hit an optimal range or a peak in the force at a certain length. And then as you lengthen it more, that force declines. Now, what is going on here? Why would length matter as far as force is concerned? So remember when we talked about myosin and actin, and we said that the more cross bridges that are formed, the more force we can produce. And so what happens is at shorter lengths, we are going to get an overlap of actin, right? And so what's going to then happen is that limits the amount of places that myosin can attach and form cross bridges with actin. As that sarcomere length increases, it reveals more spots, more places where cross bridges could form. However, if we stretch out that fiber too far, well, then you are also going to limit the amount of cross bridges that are formed. I just want to go back to this slide here to maybe show a better view of what I'm talking about. If your sarcomere is short, again, you see that these myosin heads, they can't attach to as many places here. While if they are in a more, advantageous length, then they could form more cross bridges and therefore produce more tension within those fibers. I'm going to talk more about length in a second because a lot more research has come out about fiber length and the amount of tension that a fiber can produce. But before we do that, let's just give a quick mention to velocity and say that if you're looking at the amount of force that your skeletal muscle can produce, it produces the most force right at the beginning of the movement, right? So the slowest velocity, you're going to get the most amount of strength in your movement. And so that starting point of that lift, that's when you're going to produce the most force. And as you increase velocity, the amount of force that you are producing decreases. Again, this is a lot to do with cross bridges. If you are speeding up that contraction time, well, then you are limiting the amount of cross bridges that can be formed during that time period. However, at lower velocities, you're going to get more attachment, and then you're able to produce more force during that movement. Let's go back to length tension for a second and discuss this relationship. This is how it is normally depicted, where, as you go from, this very, very small sarcomere length to around 1.8 micrometers to around 2.7 micrometers, that's called the optimal range because you're going to produce, if you just did an isometric contraction at these lengths, you're going to produce the most force here. And as you get to extending that skeletal muscle fiber and then you get less of these myosin and actin cross bridges, well then, of course, you're not going to be able to produce as much tension. The problem with this graph is it only describes this relationship in terms of isometric contraction. So setting a length and then just conducting an isometric contraction. But this relationship is not the same when you are undergoing an eccentric contraction. So, of course, we're still going to be limited as far as the tension produced by cross bridges once you start to extend the fiber and the sarcomere past a certain length. But what happens during an eccentric contraction is that you are going to get an increase in tension as you continually activate and lengthen the fiber, because a structural protein, which we talked about last time by the name of titin, is going to start to produce tension as well once we get out to these longer lengths. And that's going to add to our total tension. And that's, a lot of times we are through textbooks and things like that, talking about this sort of old methodology, or we're only looking at a snapshot of, the whole picture. So during eccentric contractions, you will get increases in fiber tension through that protein called titin. Another problem with the traditional graph of the length-tension relationship is that it doesn't account for this phenomenon that happens when you actively lengthen a fiber and then do an isometric contraction. So what would happen, let's say if we took this sarcomere at this length, and then we did an isometric contraction, but then we did an eccentric contraction, and before that muscle fiber relaxed, we did another isometric contraction. And work by Dr. Herzog showed us a really weird phenomenon that happens when you actively stretch a muscle fiber. And so here in this diagram, we have 4 different fibers. One that started at, a relatively small muscle length, and then that one that was a little bit longer, one that was a little bit longer, and then one that was at 10 millimeters, right? And so this fiber here is going to be stretched the most, right? So when you see the black line up top for force, this fiber was stretched by 10 millimeters. This fiber was stretched by, let's say, roughly 7 millimeters. This fiber was stretched by roughly 3 to 4 millimeters. And this gray one was not stretched at all, right? So these three, the black, the blue, and the orange-yellow here, all underwent an eccentric contraction. And you could see that right here. So black, blue, orange, they're going to be contracting and then lengthened here through this eccentric contraction. And then what happens is we see that the force produced by the most lengthened fiber, this black fiber here, is actually greater than the other fibers and is much greater than the one at the fiber that did not go through the eccentric contraction at all. And we call this force enhancement. And so this is a phenomenon that happens when we actively lengthen a fiber. I more so mentioned that one because a lot of times when you're coming up through undergrad and you're first starting college, you're reading these textbooks and you're learning a lot about these different things. But I just want to sort of make note that a lot of these topics like cross-bridge mechanics and force generation, there is research out there that has given us more in-depth look at these topics. And we're finding some things that we didn't know before, and this sort of changes the way we view skeletal muscle and just physiology as a whole. In order to increase the size or number of cells in the skeletal muscle or the muscle in general, we have two different methods. We have hypertrophy, which is the enlargement of existing cells, and it can occur in all types of muscle cells. And it's also going to help repair damaged tissue. So that is the goal of your resistance training, right? So when you go and lift weights and you lift heavy objects, you're trying to signal that hypertrophy. You want a cellular event to occur to increase the size of those muscle fibers so you can continue to lift more weight and be stronger, right? So that happens through hypertrophy. Hyperplasia is when you increase the number of fibers. And this doesn't happen in skeletal muscle or in cardiac muscle. So if you want to grow the size of your muscles in your skeleton, of your skeletal muscles and your cardiac muscle, you don't really create new cells. You just increase the size of the already existing ones. And in order to do that, you need to provide enough stress, enough resistance in order to signal that growth response. And that's why, if you want to build muscle, lift heavy. Similar for cardiac muscle too. If you want to increase the size and hypertrophy of those cardiac muscle fibers and you want a better cardiovascular health, you need to be participating in cardiovascular activities. And so, in order to push yourself, you could try to bump up intensity. And these are ways that you can try to signal that growth and signal that hypertrophy. That is going to conclude this lecture on force and substrate utilization. And so go back and make sure that you know the different ways where we get ATP to use for our muscle contractions. Make sure you know how we generate force and how we can generate more force. So, releasing more calcium, forming more cross bridges by, increasing action potentials, the length of the fiber, velocity of the fiber. These are all different ways that force is affected by our skeletal muscle physiology. And, just make sure that you're going through all of this. This is a lot of information. This is a really dense chapter and past few lectures here. So, if you have any questions, please let me know. But thank you guys, and I'll see you next video.